Hello, Bio 111A students. This is Doc Martin, hopefully to have a few words with you here this afternoon about the care and feeding of streaking out things to single colonies here in lab. And this is a pretty important skill for the rest of the lab, and I want to make sure that everybody's on the same page. So let's get started. For one thing, when you look at a plate, remember that this is the top of the plate. I labeled it top because it doesn't have any media in it. Now the media is the nutrients that the bacteria grow on, and they've a solidifying agent called agar, and I call that the bottom of the plate, just so you know. When you label a plate, do be careful. Do you notice that you want to write the name, your lab section, the date, and some form of identification? For example, in this case, maybe it's your water bottle buddy number 1. Let me explain what I mean by that. For this part of the course, it's really important for you each to have 2 isolates from your water bottles that have been streaked out to purity, forming single colonies that are separate from one another. Each one of you are going to get 2 that you've made. Now, for some people, that's very easy because of the amount of material that they had in their water bottles, and for other people, it's more challenging. But that's why we're going to spend this next week in lab really focusing on getting it done. Let me show you what I mean. For some people, and it's really important to understand I'm not singling anyone out with this, here are some single colonies that are well separated and can be gotten individually. Remember, if you don't have single colonies where you can do that, you're going to have real trouble when we're doing PCR and identification because there would be more than one type of bacterium involved. The single colony means there's one type. Now, some of you have more challenges. For example, if you look at this plate, you'll see that there are at least 3 different types of colonies here, maybe 4. There's a large pale one, there's small yellow, small white, and maybe a small pale one. And that's why we're going to spend some time in lab talking about it. For other people, things are a little bit more challenging as well. If you look at this, actually there are some single colonies here at one edge that you could get to, which is good news. But generally speaking, the streaking here was not as I would like you to do it, although it looks as if this one worked out, which is great news. Here's an example of where there's extra work necessary. As you can see here, there are no single colonies. How did that happen? And I think the best way to explain that is to go over how single colonies work, or how streaking to single colonies works. So in order to do that, I have to angle up the burner a little bit. I want you to notice that this is the inoculating loop that everyone needs to use right there. And there's actually a blue cone of flame that ends about here. Do you see how quickly that heats up? And when I walk it all the way down, I get everything nice and sterile when I do that. Now, I want to warn you, if you just leave it low, do you notice how the center doesn't heat up immediately? That's because it's too low. Go a little bit higher up and it heats very, very well. Now, I also want to warn you that sometimes when you come to lab, you're going to see like a kink or a bend in your wire loop, and you'll want to straighten that out with your fingers, but make sure it's not hot when you do it, because hot wire looks a whole lot like cold wire. And I have done that more than once, where I branded myself essentially. So I'm going to try my best to show you what I have in mind with a plate. And there are 2 ways to do the streaking, and I'm going to show you both of them. So what I want to tell you about is you go ahead and you get your, as I told you, let's turn it up a little bit so you can see. You want to get your loop nice and hot. And I walk that down there. And it's going to start to cool as soon as I stop, yes. And then what I do is I come over to a plate. Now remember, this tip may be hot, so I touch it to a part of the auger I haven't used, and that makes sure it's cool. And then I get right over here a tiny bit of a colony. Do you see it on the end of the loop? You probably can't, but it's there. You can tell because I kind of mutilated the colony. I want to warn you that every colony that you've seen thus far has well over 1 million cells. Now I want you to then take what you just picked up with your loop, and you obviously don't sterilize it at that point because it picked up some cells. Do you see how I used both sides of the loop and made a little tiny mark with it? And if I go back and forth a little bit with the light, you should be able, there we go, to see where I did that. I then heat up the loop all the way down the wire, just like we've talked about. I touch it to a part of the plate I haven't used before. Now this next part's really important. What I do here is I go once through what I did, and then not back through it. I instead go back and forth and back and forth and back and forth many times. I'm diluting the number of cells on the loop when I'm doing that because they're getting rubbed off. And if you go back and forth in the light, you can see where that is by the reflection of the light on the surface of the plate. Now you need to re-sterilize the loop, which you do. Again, go to a part of the plate that you're not using and cool it. Turn the plate so that you can see from the kind of streak marks where things have been, and you go once through that area, just once, and then not again. Do you see how I'm doing the same thing I did in the first sector, where more and more cells are being rubbed off? I'm not going back through what I did before, however. And now I go and I re-sterilize the plate, just like before. I turn the plate and I make sure that I can see what I did before, and I am now looking at what I had done before. And then I go once through that area, and again, not back through it again. And that way more and more material has been rubbed off. Now, and then you want to sterilize your loop when you're done, of course. Now, I want to tell you something important. That first thing that I did, that first bit right up here, that first bit, had a lot of bacteria. And I went once through that area and then back and forth many, many times moving down the plate. That rubbed off more and more cells. Then I sterilized the loop. I went through once this material I had just made, gone back and forth with many times. I went once through that area and then back and forth many more times. So what I'm doing here is I'm diluting. I guess that's a funny word to use with a loop. But there are fewer and fewer cells on that loop because they're being rubbed off. And then finally that last one. And again, just like before, and then you can see it there in the reflections. So generally down in here where I'm pointing, you're going to have individual cells, and what they're going to do is they're going to grow into individual colonies like you see there. And that's the goal. So I hope that helped with that aspect of it. The other thing that I wanted to say is sometimes you have a really complicated looking plate, a plate, for example, where you have lots of different looking things, and it's kind of hard to figure out how to get to that with a loop. Maybe if you just took a small yellow colony, that's possible. Maybe if you took one of the whitish colonies, you could do that. But you can see there's a lot going on there. How do you go about doing that? Well, in that case, I would use sterile yellow tips rather than a loop. And this is what I would do. And remember, I have a plate ready when I do this. Now, when I pull out one of these, that part there is sterile, does not need to be flamed. And then I get my plate and I say, all right, what am I going to go for here? And I want something that's kind of well isolated. How about this one right here? And then I touch the tip to it. And you can see how I mangled that colony a little bit, and that means it's on the tip. And then I get myself a new plate that's already pre-labeled. And I want you to notice that I smear that material in a small area, and I'm rotating the tip as I do it to make sure the material is there. Now this gets thrown in biohazard, of course. Now what I do is I get things nice and heated up with my loupe. And just like I did when I used a loupe initially, I go once through the area I put on the plate, so I need to identify that again, and I see it right there. I go once through that area, let me get this where it makes sense to you, and I don't go back through it, and if I tear, I just turn it a little bit and hold it nice and lightly. And that means there are fewer and fewer cells there. Now I go ahead and heat my loupe again. And then I turn it. I cool the loupe where I haven't gotten bacteria yet. I go once through the area in question and back and forth many, many times. And then I go and I re-sterilize the loupe a final time. If I don't re-sterilize the loupe, I'm just smearing the same amount of bacteria around, which is not how you get single colonies. And then finally, and let's make sure you can see them here. There you go. On the screen, you can see where I've done stuff before. Cool the loupe, go once through, and not back through what you did, mind you. And then you're done. Now, some people do 4 quadrants, and I generally do 3 because I was never a streak plate artist, as the saying goes. But I want to remind you about what we did. I want to tell you that we had that initial, where my finger is right now, tap, tap, tap. That was a small amount of material from the yellow loupe, and the yellow tip that I took from a colony on a plate. And then, using my sterile loupe, I made sure it was sterile. I went once through that area and then back and forth many, many times. Sterilize the loupe. Go once through that first sector, as I think you can see, there it is, many, many times. Re-sterilize the loupe, and then back to the final sector. Again, the idea is to have fewer and fewer cells over here as you go through the sectors, so that you have individual cells down here which grow into individual colonies. And that is how you deal with a complicated plate of that nature. Which brings to mind, you can see how you could use this approach to pick individual colonies here, perhaps using a yellow loupe. That would work. I could see that. The question becomes, what do you do with a plate like this, where there really aren't single colonies? And all you can really do here is take a yellow tip, and you want to get something from the very edge and then try and streak it out. And that would be my advice to you. What I've done is I've made extra R2A plates over the weekend, and so you should be able to do that this week, and I'll talk in lab about what everybody needs to do record-wise, because it's very important that you know what kind of isolates you have. For example, the student who has this kind of colony isolate, this might be their water bottle buddy number 1. We could use these colonies right now to do the PCR experiment and identification, but we would need to know it was their first isolate and that it appeared to be yellow and maybe has a photograph associated with it. More about that in lab. So this is the kind of record keeping that you'll need to do for class. And I'm really happy to have you in this class. And again, what we're doing in this course-based undergraduate research experience is something that's genuinely important. The search for new bacteria or other microbes that produce antibiotics. Some of you have found them from your water bottle isolates, but most of you will find them from the soil. So thank you so much for your time, and I really appreciate you watching this video and thinking about it. I hope you have a wonderful Sunday. Bye.